Hello, my name is Wali Agamas. I am an infectious disease physician and currently a preventive medicine fellow at Tufts in Maine Medical Center. I'm going to talk about COVID-19 vaccine. The lecture is for medical students and the material is up to date as of March 2021. So I will start with the basics by defining COVID-19. The word COVID represents coronavirus disease that was first noted in 2019. The World Health Organization came up with that name. So it is a disease caused by a type of coronavirus called SARS-CoV-2, which is also an acronym that represents Severe Acute Respiratory Syndrome Coronavirus 2. It was first detected at the end of 2019 and soon declared to be a pandemic. So it is a deadly and highly contagious infection. Due to limited treatment options available at this time, prevention is the best alternative. Speaking of prevention, vaccines are among the best preventive means we have. In order to understand how the vaccines are made and how they work, it is very important to know the different viral structures. The picture projected here is SARS-CoV-2 virus. The virus has many structural parts, but the most important ones are the ones labeled here, which are the surface proteins that include the spike protein and the M protein, and the viral genetic material, that's the RNA. The spike proteins are especially important because the vaccines are made targeting the spike proteins. They are crown-like structures that are found on the surface of the virus. By the way, the name corona came from these crown-like structures. The progress in producing vaccines against COVID-19 is very impressive. More than 90 vaccine candidates have been in development by different research institutions and universities across the world. Various technologies are used. Some of them haven't been used in a licensed vaccine before. At least 6 groups have started injecting, and 3 of them, namely the Pfizer, Moderna, and the Johnson & Johnson vaccines, have received their emergency use authorization in the United States. So what are the types of COVID-19 vaccines? There are 4 types of vaccines based on how they are made. Those are the viral vaccines, the nucleic acid vaccines, viral vector vaccines, and protein-based vaccines. We will discuss in detail about each of them in the next few slides. Of the 4 types, nucleic acid-based vaccines have not been used before, but have been studied since the 1990s to make vaccines for other viral infections, such as SARS. So, the technology is not brand new. But irrespective of the type of vaccines or the methodology used, all vaccines aim to expose the body to an antigen that will provoke an immune response by the host system. Now let's discuss about each of the vaccine groups. The first one is the viral vaccine. These types of vaccines use killed or inactivated virus or live but weakened virus to make vaccines. At least 7 teams are working to develop vaccines using this method. Sinovac BioNTech from Beijing is one of those groups testing COVID-19 vaccines using this method. From the existing vaccines, measles and polio vaccines are made using this technology. These types of vaccines require extensive safety testing, especially if weakened live vaccines are used, because you don't want to cause an infection while trying to prevent them. The second group are the nucleic acid vaccines. This includes the RNA and DNA-based vaccines. At least 20 teams are using this technology to make COVID-19 vaccines. This technology is relatively new, but easier to make vaccines. That is because to produce a vaccine using this method, It involves making genetic material only, not the whole virus. And then the nucleic acid inserted into the human cells uses the cell protein synthesis machinery to make copies of the virus proteins, in this case, the spike proteins. This is a diagrammatic representation showing how the nucleic acid vaccines work. The genetic materials, that is the DNA and the RNA that codes the target protein, in this case the spike protein, is engineered from outside and given as a vaccine. Once injected into the human body, it will go into the cell. The DNA then will go into the cell nucleus here and transcribed into messenger RNA. Note that even though it gets into the nucleus, it is not incorporated into the human genome, and it doesn't affect the genomic composition of that individual. On the other hand, the current RNA vaccines are messenger RNA, and they don't need to go into the nucleus for transcription. Then the messenger RNAs then use the cell machinery to produce the spike proteins, and those spike proteins provoke an immune response by the host system. The third group are viral vector vaccines. A virus such as adenovirus is genetically engineered so that it can produce coronavirus proteins in the body. These viruses are, the vector viruses are replication incompetent and they cannot cause disease. Around 25 groups are working on this. This is a diagrammatic representation showing how the viral vector vaccines work. So this is the coronavirus, and the red portion is the genetic segment that codes the spike protein. This genetic segment is cut from the coronavirus and placed into the vector virus, in this case the adenovirus, and then given as an injection as a vaccine. After injection, it will go into the human cell and spike proteins will be produced. That again provokes an immune response by the host. The Johnson & Johnson and AstraZeneca vaccines use this technology. Johnson & Johnson uses replication incompetent adenovirus type 26 or AD26, while the AstraZeneca vaccine uses chimpanzee adenovirus. Note that these viral vectors do not cause any infection or other diseases. The fourth types are protein-based vaccines. This is a method of provoking immune response by integrating or injecting fragments of proteins or protein shells that mimic the coronavirus outer coat. Several other vaccines are made using this technology. This type of vaccines need an adjuvant molecule for adequate immune response. Now let's talk about vaccine development. The whole process of vaccine development takes up to 10 to 15 years. The current COVID vaccine took only about a year. That's because, as I have mentioned earlier, the technology has been studied for more than 20 years. This vaccine development process has multiple phases. That includes the clinical and the preclinical phase. In the preclinical phase, vaccine candidates are tested on test animals. And in the clinical phase, in phase 1, safety and dose ranging will be tested. The safety of the vaccine candidate with different dose range will be evaluated using healthy volunteers. A small number of participants, usually in the tens, are required during this phase. In phase 2, immunogenicity, that is the capacity of the candidate vaccine to cause immunity to provoke immunity is tested. In this phase, patients or target populations that are intended to be used after approval needs to be included in this phase, and usually hundreds of individuals are needed. The third phase is the phase where primarily tests efficacy, but safety is already there as well. A large sample size, usually in 10,000, are needed in this phase. Once phase 3 is completed, the next step is submission of application for approval, usually to FDA. After approval, there are post-approval studies. Usually these kind of studies are comparative studies. For example, comparing the newly approved vaccine with the old vaccine, if it exists one before, or new population can be studied. For example, for the COVID vaccines, children, pregnant women, and immunocompromised individuals were not included in the vaccine trials. So those can be the population to be studied in the post-approval phase. Another area is post-approval surveillance. VAERS is the best example, which represents Vaccine Adverse Events Reporting System. It's a federal database where vaccine adverse events are reported, and it provides very valuable safety data. So what is an emergency use authorization? It is a mechanism to facilitate the availability and use of vaccines or medication or diagnostics during emergency situations before they are officially approved. EUS allow the use of unapproved medical products. Example, COVID vaccine has been allowed to be used while phase 3 trials are still ongoing. EUA can also allow unapproved use of approved medical products. If you recall, hydroxychloroquine was used to treat COVID infection at the beginning. Even though hydroxychloroquine is an old drug and has been in use for several years to treat malaria and autoimmune disorders, it was not initially approved to be used for COVID. So in order to use this medication to treat COVID, it needs an emergency use authorization. As of March 2021, 3 vaccines, that's the Pfizer vaccine, the Moderna vaccine, and the Johnson & Johnson vaccines, have received their emergency use authorization in the United States. The details about each vaccine is discussed in another video. These are the references. I really encourage you to visit the CDC website, especially the clinical consideration section. That's it. Thank you very much.